The next one is a cytokinin. So cytokinin, we know that cytokinins, they were discovered in the cells for the factor that stimulate the plant cell to divide. And at the name suggests, the cyto means the cells and the kinins means that to divide, it undergo the division. So a great many substances, they were tested in an effort to initiate and sustain in the proliferation of the normal stem tissues in the culture. The material ranging from the yeast extract to the tomato juice, they were found to have the positive effect. Okay. However, in the vitro tissue culture, growth was stimulated most dramatically when the liquid endosperm of the coconut was added to the culture medium. So cytokinins are the class of the plant hormones that play a very crucial role in the various physiological process, including the cell division, the shoot and the root growth, the chloroplast development, the delay of the senescence. Okay, it's a delay of senescence means delay of the aging and the regulation of the nutrient metabolism. So let's discuss some key points about this cytokinin. So obviously they are involved in the cell division because it promotes the cell division in the plant tissues, particularly in the meristematic cells or meristematic region, leading to the overall plant growth and the development. Next is in the root and the shoot growth because they stimulate them by promoting the developmental of the lateral buds, okay, and preventing the apical dominance. Okay, they also influence the root growth and the branching. Next is the chloroplast development. The cytokinins, they regulate the chloroplast development and photosynthesis by influencing the chloroplast size, number as well as functions. Next is in the delay of the senescence. So cytokinins, they delay the senescence, the aging process in the plant by inhibiting the breakdown of the chlorophyll and the other cellular components. So in this way, the delay of the senescence occurs. And in the nutrient mobilization, they influence the transport of the nutrient within the plant, particularly the nitrogen compound. Next, they are helpful in interaction with the other hormones also. Like the cytokines, they interact with the other hormones such as the auxins, gibberellins, and abscisic acid. Okay, to coordinate the various aspects of the plant growth and the development. The biosynthesis, they are particularly synthesized in the root, but they are also produced in some other plant tissues also. The biosynthetic pathway, they involve the conversions of this adenine nucleotide to the bioactive cytokine. Next is in the transport, so they can be transported throughout the plant via the xylem and the phloem, allowing them to exert the systematic effect on the growth and the development. So the common cytokinins type, they include the zeatin, the isopentenyl adenine, that is IPA, the dihydrozeatin, and the applications. Even some free forms of the cytokinins are available, which includes the ribosides, the nucleotide, all this. So applications, they're involved in the cell division. They are also involved in the various purposes in the agriculture, like in the cell division, enhancing the shoot growth, the dealing analysis, improving the crop yield as well as the quality. So the discovery of this cytokinin was, done by the Folk-Skoog and the Kovac, or we can say the Koog and the Miller. Okay, so in the 1940s and the 1950s, the researchers of the University of the Wisconsin, which is led by the Falk Scoop, they tested the various substances for their ability to induce the cell proliferation in the culture tobacco pit tissue. And then the observation of the adenine effect, that initial experiment shows that the adenine a nucleic acid base had a slight promotive effect on the cell division. Okay, on the cell division. So this experiment showed this result. So the discovery of the kinetin, surprisingly, the aged or the autoclaved herring sperm DNA, which contain the adenine, they exhibit a very powerful cell division promoting effect. And further investigation, it led to the identification of the small molecules from the autoclaved DNA, which is known as the kinetin. So this led to the discovery of the kinetin. Then some chemical structure were identified because the kinetin, they were identified as the adenine derivative, which is known as the 6-furfuryl aminopurine. Okay, aminopurine. So it is not a naturally occurring plant growth regulator and also not found as a base in the DNA of any species. So the origin of the kinetin is basically a byproduct of the heat-induced degradations of the DNA, and its discovery suggested that the presence of the naturally occurring molecule with a structure similar to the kinetin. This is the structure of the kinetin we can see. So, their importance in the plant biology or the discovery of the kinetin, they provide insight into the regulation of the cell division in the plant, and they pave the way for the further research into the plant growth regulators and their role in the plant development and the physiology. So these are the cytokinins. So naturally occurring cytokinins are all the adenine derivatives with an uh, either with the isoprene related chains or the aromatic cyclic chain. So the former are basically known as the isoprenoid cytokinin because they have the isoprene related side chain like this. Okay, and although there are some variation also depending upon the species and the developmental stage, so most of the common isoprenide cytokinins are the N6 isopentenyl adenine, that is the IP. Okay, then the dihydrozeatin is there, that is DZ is there. The transzeatin is there, which can be converted into this ciszeatin. Okay, so these are the naturally occurring cytokinins. The aromatic cytokinins such as the benzyl adenine, that is the BA cytokinin, they are the less common. It is found in very low species. So this zeatin, as we are watching here, so zeatin is the most abundant naturally occurring free cytokine, and its molecular structure is similar to that of the kinetin. Although they have the different side chain in both the side chain, it is linked to the nitrogen. As we can see, it is linked to the uh, nitrogen, which is attached to the C6 is equals to the N6 of the adenine. Okay. Now, because of the side chain of the zeatin, they have the double bond. As we can see that this zeatin, they have the side chain which has the double bond. Okay, so they can exist both in the trans form as well as in the cis form or the cis configuration or the trans configuration. Since its discovery and immature,